Vision can be said to be the most important sense for the human experience, and much of our brain is dedicated to processing visual information. In these pictures, there are just a few aspects of the visual system that I want to talk about, or the visual sensation that I want to talk about. There's much more to be said, especially in terms of central processing, that I'm not going to get into. We know a lot about vision because extensive research has been done, especially in the 1960s, on the neurophysiology of vision and how that's processed. It's really quite a fascinating system, and so much is known about it, it could make up an entire semester course all by itself, I'm sure. In fact, I have a textbook in my collection that's essentially just about how vision processing works. This is a horizontal section through the eyeball showing its different regions. The main part that I want you to understand about the structure of the eye, and there's more detail for this course presented elsewhere, is the idea of the eye as a camera. Or really, truthfully, the camera is just a recapitulation of what the eye is. Anatomically, probably the most important aspect of that to assess is the anterior and posterior cavities that are depicted here. The anterior cavity is where the lens is, as well as a number of other structures that control the entry of light into the eyeball. And the lens, of course, is important for focusing that light onto the rear portion of the eyeball. In the posterior cavity, which is mostly filled with the gelatinous goo of the vitreous humor that light goes through, mostly unimpeded. Most importantly, though, for transduction of vision is the retina, the actual neural component of the eyeball. And the lens is focusing light onto the eye. And the most important part, you could say, perhaps, is the region labeled here as the fovea centralis, often just referred to as the fovea. That's essentially the exact center point of the retina. When we are focusing on something, perceiving the very middle of our visual field, what we're doing is we're processing the stimuli that are falling directly on the fovea. You can notice in this picture it's a little bit of a pit right there. The retina is a little bit thinner just so that light can fall on the photoreceptors most directly. The other components have moved out of the way. Not that they physically move every time we're using our eyes, but the structure of the retina is such that the other layers of the retina are not impeding the path of light to the photoreceptors. And I'll show you the structure of the retina in a second. But that's the fovea. Surrounding the fovea, we have the region called the macula, which you may have heard of in terms of macular degeneration. The macula is just central visual field. Fovea is the exact center where we're looking at, and the macula is generally the area that we can imagine. So think about, if you stare at the middle of this picture, the very center of the picture of the eye, you're looking at one point, and that's where the fovea is noticed. If you don't move your eye, but you consider that you can see the rest of the picture of the eye, that's what the macula is processing. Again, without moving your eye off this picture, consider where the text is, and that you can't make any sense of it. You can't read it that far out in the periphery. That's beyond the macula. We just don't get enough visual information to get any detail. We do know it's text because of its general appearance, but you'd have to look again directly at the text to be able to read it, which would be moving the image of the text onto your fovea, which you can see there. The retina is made up of a highly organized set of layers of cells. The outermost layer furthest from the center of the eye are the photoreceptors. Now, these are going to be furthest from the light source, since the retina does not go all the way around the eye, but only around the inside of the posterior cavity. And so light has to pass through the other layers in order to get to the photoreceptors. As I was talking about the fovea a second ago, the other layers are not in the light path going to the photoreceptors. There are two types of photoreceptors. There are cone photoreceptors and rod photoreceptors. Rod photoreceptors are specialized for activation by low light levels, so we associate those with visual stimuli in dark settings. If you're looking around your bedroom at night with the lights off and maybe only some moonlight shining through the occasional window, then your rods are largely the only thing responsible for visual sensations. Cones are specialized for color perception. There are 3 different types of cones, being able to differentiate different wavelengths of light or different colors. And our brain can take the data from those 3 types of cones and establish a nearly infinite number of colors that we can perceive. How many we actually are able to name depends on our experience with color vision, but it's a very sensitive system to get fine detail in the shades of color that we might experience. They, however, are not sensitive to low light situations, so in that dark room setting I just described, the cones are effectively turned off, or they're not responsive to light. However, when we're looking under normal lighting conditions on a clear day, for instance, there's so much light, the rods are not going to be active. They'll be bleached out by all that light, and the cones will be really the only component of our visual system. And that also goes to explain, if you've ever noticed, that you don't really see colors in the dark room at night. You only see the colors during the day. If you think you see colors at night, actually you don't. Any perception you have of color probably comes from your experience. You know what color the walls in your bedroom are painted or something like that. If you think about the nighttime bedroom scenario I suggested, usually the brightest light somebody's going to have is going to be the clock face they have if you have a digital clock, which, depending on the manufacturer, might be made of red light or blue light. There's different reasons to use different colors that I'm not going to go into, but those are going to be bright enough that the cones can differentiate the color, but everything else is going to be so lightly, I shouldn't say lightly, so lowly illuminated that only the rods are going to react to light coming from them. The other layers of cells, moving inward from the photoreceptors, we go to the bipolar cells, named strictly for their shape, and they are just relay cells between the photoreceptors and the other layer that I'm about to describe. The innermost layer, the one closest to the light source, is made up of what are called retinal ganglion cells, or RGCs sometimes. Those cells are receiving the information from photoreceptors through the bipolar cells, and the retinal ganglion cells then send that information into the central nervous system. Axons of the retinal ganglion cells make up the optic nerve, the second cranial nerve, and they line actually the inside surface of the retina. So light has to go through those axons as well, and they collect at the optic disc and emerge from the eyeball as the optic nerve, which then enters into the central nervous system, where it then becomes called the optic tract, and it synapses in the diencephalon. And then our visual perceptions and stimulation is based on diencephalic activation. The thalamus primarily is dealing with our conscious perception of vision, and the hypothalamus and midbrain are dealing with our unconscious perceptions. The hypothalamus dealing with vision, helping to lead into homeostatic mechanisms, and the midbrain involved in visual input to motor functions that are not necessarily consciously perceived. There is an odd situation where somebody might be blind but can still react to visual stimuli. If blindness, the removal of conscious visual perception, is taking place at the cortical level in the cerebrum, then nothing is wrong with the visual system between the eye and the hypothalamus and midbrain. And so visual stimuli can be sensed, you could say, but we won't consciously be aware of them. So you might, don't try this. I'm not suggesting that you do this at home or anything like that. But if somebody has this sort of blindness situation, they might not be able to see you throwing a ball at their head, but they would be able to catch it or deflect it, because the motor component through the midbrain, for instance, would react to the stimulus, even though that they weren't consciously aware of the ball coming at them in a visual sense. So there are some quirks of how the visual system is set up and wired that you can have visual activity at a subconscious level but not be consciously aware of it because blindness at the cerebral level. But that's beyond the scope of this course. I just wanted to suggest that there are multiple visual processing streams in the central nervous system, some subconscious and some conscious. As I said earlier, the visual sense is very well described, and I could spend a lot of time going into a lot of aspects of what's going on. My description of the retina just a second ago left quite a bit out, and I don't need to get into a whole lot of detail of that. A little bit more is covered in the book, and you can certainly find sources elsewhere that go into more detail, but that's not really what I want to stress. And just to give you an idea about the transduction mechanism of vision, I want to talk a little bit about that. This is a picture of what's happening within a photoreceptor. This is showing us a rod. A very, very similar process would be taking place in a cone. In the outer portion of the rod photoreceptor, which again is the furthest from the light source through the retina. So light has passed through the blood vessels on the inside of the retina, and through the axons of the retinal ganglion cells, through the cell bodies of the retinal ganglion cells, through the synaptic layer of the retinal ganglion cells and the bipolar cells, through the cell bodies of the bipolar cells, through the synaptic layer of the bipolar cells and the photoreceptors, through the cell bodies of the photoreceptors, called the inner segment, and finally the light reaches the outer segment of the photoreceptor, and that's where this process is taking place. There are stacks of membrane discs within the rods, not exactly stacks of membrane discs in the cone, but still a similar arrangement. And in the membrane of these discs are the photoreceptive proteins called opsins, and they contain within them a specialized molecule called retinal, which is derived from a vitamin that we get in our nutrient load in our eating. When a photon hits this molecule, it chemically changes. Before being exposed to light, it's a molecule called retinal, and it is chemically modified into cis-retinol. And trans and cis are just describing different aspects of how carbon molecules are bound together. And it changes the shape of the chain within this molecule. When that molecule changes shape, the opsin protein reacts to that, which leads to chemical changes within the cell, which then leads to membrane potential changes eventually as ions cross the cell membrane. And that's going to lead to neurotransmitter release at the synapse with the bipolar cell, and then the bipolar cell will generate an action potential activating the retinal ganglion cell, which then will send an action potential down the optic nerve and into the brain to elicit visual perception. This is just a quick idea of what's happening way down at the molecular level. I just want you to understand that it can be a very complex system, and there's a lot going on. I'm not going to ask a whole lot of deep questions about this. I just want you to be aware of this level of activity. Also notice the cyclical nature of what's described here. The retinol molecule is modified by the photon, and when in that form, the opsin is non-responsive to light until it gets recycled back to the original form. When opsin's not active, that's described as being bleached, and it has to recover. You're familiar with this probably if you're ever exposed to a bright light source. The photoreceptors that are exposed to that are bleached temporarily. If you stare at a light for a little while, close your eyelids, you will see an after image of that, which is actually the absence of activity of those photoreceptors, and in comparison to the base activity of photoreceptors that are not bleached, gives you that sort of negative after image. Once they've been recycled and they're active again, the after image will go away. But that's happening, again, at the chemical level within the photoreceptor, and it's essentially the basis of what causes vision. It gets translated up to our experience because of the transduction mechanism, which is actually suggested here and gives rise to the membrane potential changes I just described. This picture indicates the structures associated with the ear. There are three main regions: the external ear, the middle ear, and the inner ear. And there are other sources, again, within this course that describe this in more detail. I just wanted to point this out here. The transduction mechanisms of hearing take place within the inner ear, specifically within the cochlea of the inner ear. And I want to concentrate on that a little bit. Without going into the structure of the inner ear too much right here, we're going to jump right to the neurons that are responsible for this, because what's important about the transduction of hearing is the fact that it is a mechanical sense. The vibrations that are established by sound waves cause changes in cells that will then transduce the signal into a action potential code in our nervous system that will sense. The mechanoreceptors that are hair cells in the inner ear have on their apical surface an array of membrane extensions collectively called the stereocilia, which is indicated in the first panel in this picture. The second panel, labeled B, shows an electron micrograph image of two of the components of the stereocilia array side by side. And hopefully you can see that there is a very fine filament connecting one to the other in panel B. Panel C kind of shows a cartoon image of what the stereocilia look like. There are two depictions superimposed on each other. The one indicated by the negative sign would be the position the stereocilia would be in when they're not active or activating, and the positive sign shows the activated position. And mechanically, the stereocilia array moves. Now, the stereocilia array, as you can see, has graduated sizes of these extensions, and when it moves towards the highest of those extensions, the tallest of them. If it moves in any other direction, what I'm about to describe wouldn't have any real effect. Now, in panel D is a cartoon sort of depicting what panel B is trying to represent. What's happening here, and this is the basis of the mechanical sense, is that there's a tether, a fine protein filament going from the taller stereociliar component down to the lower one, connecting them. And where it's attached to the lower one, there are ion channels. And if these two components move towards the taller one, tension will be put on the filament represented by the sort of two lines that are intertwined around each other in this picture. Tension will be put on that, and it will physically pull on the channels, open them up, and allow for ions to cross the membrane of the stereocilia, which is really the membrane of the whole hair cell, resulting in membrane potential changes and therefore transducing the vibrations of sound into changes in membrane potential and the action potential signal that will be traveling along the vestibulocochlear nerve. Now, this is a very mechanical reaction, and I'm just pointing out here the exact mechanism of transduction from the physical movement of these stereocilia to the opening of ion channels. There's quite a bit more to that. There's a video that you'll find on the Blackboard website that you can watch about how this transduces in a bit more detail, but this is to just get at the idea of how transduction takes place at the same level of what it described for taste. So, sodium is rushing into a cell, depolarizing it, is essentially what's happening here. Originally, I started talking about the senses as the big five that you might hear from pretty much anybody around. There is a sixth sense in terms of that list of five that we talked about, which, remember, is not really an exhaustive list. There's many, many more senses, often submodalities of those big five. But there's other senses that I want to talk about, and it's not the sense of seeing dead people. The real sixth sense is our sense of equilibrium or balance. If you think about the five, touch, taste, smell, sight, sound, hearing, vision, however you want to describe those last two, it doesn't include our sense of balance or equilibrium. Possibly it doesn't make it into those list of five because equilibrium is handled by the inner ear with hearing. So you could say that it's not in that list by itself because it could be considered a subset of hearing. So not really, it's a very different sense in what it's processing stimulus-wise. However, it's anatomically very similar because of where it is and what cells are involved in transducing the signal, which in fact is still the same kind of hair cells I just described for the sense of hearing. The sense of balance or equilibrium is really two different things. There's the sense of head position in a static sense, how your head is sitting relative to the environment around it, and the sense of head movement. So this picture depicts how head position is sensed. It's sensed through two different regions within the vestibular part of the inner ear. The snail shell-looking portion is the cochlea. The other portion, which is highlighted in this picture with the two blue pieces, and then we're about to look at the three sort of circular or semicircular canals off to the side. The two blue things here, the utricle and saccule, are involved in head position. One specialized for vertical position, the other specialized for horizontal position. By putting those two planes together, our brain can sense our head position in many different planes of orientation. But one strictly horizontal, the other one strictly 90 degrees opposite that. Off to the side, we see what one of these looks like, and this would be what's probably dealing with horizontal position based on the drawings here. There is a support layer of cells, referred to as the macula in this picture, into which hair cells are embedded. The stereocilia of the hair cells are extending out into a gelatinous goo region where some mineral crystals are found, the otoliths. The otoliths directly translate as ear rocks, odo for ear, lith for rock. And being calcium crystals, they are essentially the kind of thing that you find in a lot of rock structures. They're denser than the gelatinous goo that they're found in, and they're denser than most of the structures that make up cellular components of our bodies. And being more dense, they're going to be acted on slightly differently by the force of gravity. So if the head's tilted forward, as this picture suggests, the otoliths will pull the gelatinous goo and the stereocilia in a particular direction. And by activating the stereocilia, again using that same tether transduction mechanism I just showed you a little bit ago, that will activate these cells, and our brains will be able to sense our head has changed its position. Now, something very similar to this is happening in a 90-degree orientation from this, a vertical plane. When the head's upright, like in this picture, that vertical plane is completely activated. As the head tilts, the vertical plane becomes slightly less activated. And so the increase in activation of the horizontal plane and the decrease in activation of the vertical plane add up together to give your brain an understanding of the actual position of your head. The utricle and saccule that I just described are sensing the static position of the head. The time it takes for those otoliths to displace the position of the stereocilia has a little bit of lag in it. So real-time reporting of the movement of the head would not be terribly accurate with the utricle and saccule. They're more for the static position of the head. For real-time movement of the head, we have instead the semicircular canals, the three rings that we see. Now you have an inner ear on either side of your head, so the three semicircular canals in one inner ear correspond to the three semicircular canals in the other inner ear. One of those canals in either inner ear is in horizontal orientation, so the two of them together define a horizontal plane of your head. The other two are in a vertical plane, one 45 degrees slightly off center midline in an anterior position, and the other one about 45 degrees off center midline in a posterior position. The posterior canal on one side and the anterior canal on the other side define a vertical plane at about 45 degree angle to midline, say from front right to rear left, and the other pair would define a 45 degree angle in the other direction relative to midline. Those three planes together make up a three-dimensional Cartesian coordinate system, if you will, defining movement of the head in real time. What happens when the head's moving is the stereocilia of hair cells located in the ampulla-cupula structure that you see here bend when fluid within the semicircular canals move. When your head rotates to the left, for instance, the fluid in one semicircular canal will slosh or rotate, if you will, in a clockwise direction, and the fluid in the opposite semicircular canal will slosh in the counterclockwise position. One of those movements will cause activation of the stereocilia, and the other will cause inactivation of the stereocilia. So your brain will know because one side's active and the other side's not active that your head is rotating in a particular direction. If you rotate your head in the opposite direction, then the opposite set of activity will happen, which will code for rotation in that way. Considering all 6 semicircular canals together, your brain can get a very accurate indication of what direction your head's moving at any particular time. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer.